In this activity, you will examine a longitudinal section of a Coleus stem tip to observe the region where stem elongation occurs. You will identify the apical meristem and the early tissue systems that give rise to the epidermis, ground tissues, and vascular tissues. Plant stems increase in length through cell division at the apical meristem, a region of actively dividing undifferentiated cells located at the tip of the stem. Those cells undergo mitosis and provide the raw material for all primary tissues of the stem. This continuous division and differentiation pushes the stem upward, allowing the plant to reach sunlight and expand into new space for leaves and flowers. Just behind the apical meristem is the subapical meristem, where cells begin to differentiate into three primary tissue systems. The protoderm forms the future epidermis. The ground meristem forms the cortex and pith. The provascular tissue, also called the procambium, gives rise to xylem and phloem. Also visible near the apical meristem are leaf primordia, which are the earliest stages of leaf development. In the angle between each leaf primordium and the stem is an axillary bud, which contains a meristem that can later develop into a branch or flower. Before observing the slide, answer this pre-lab question. Why does the apical meristem play such a critical role in stem elongation? Consider why growth in length must be restricted to a specific region rather than occurring throughout the stem. Think about where new cells are produced and how those cells contribute to tissue formation. Go ahead and pause the video to write your prediction in your lab notebook, then continue. Here is the stem tip of a Coleus plant. Under this power, locate the very tip of the stem. Identify the dense region of small, tightly packed cells that marks the shoot apical meristem. Look for nearby leaf primordia and developing tissue zones. The leaf primordia are tiny horn-like or finger-like projections that curve over the shoot apical meristem. You will have different stages of development. These lack the full differentiation of tissues like vascular bundles, mesophyll layers, and epidermis with stomata that its mature counterparts have. It also has not expanded to perform photosynthesis yet. You can also see trichomes or hair-like structures visible along the outer edges of the developing leaves. Just behind the apical meristem, identify the three primary meristems. The protoderm is the outermost layer of cells covering the shoot apex and leaf primordia. It is a single layer of cells that will eventually mature into the epidermis. The provascular tissue, or procambium, is visible as dark elongated strands of cells running vertically through the center of the stem and into the developing leaf primordia. These cells are currently undifferentiated vascular tissue that will mature into primary xylem and phloem. The ground meristem is tissue located between the protoderm and the procambium, as well as in the very center of the stem. It fills the bulk of the stem and will differentiate into pith and cortex. The axillary bud is a small, dark lump of meristematic tissue located in the axils, where the leaf attaches to the stem. It has the potential to develop into a lateral branch, a cluster of leaves, or a flower. Find a region where a developing leaf attaches to the stem. Observe how vascular tissue diverges into the leaf as a leaf trace, leaving a temporary leaf gap in the stem's vascular column. The leaf trace appears as a dark elongated strand of procambium. The leaf gap is ground tissue directly above the point where the leaf trace departs. Pause and draw the embryonic regions at the stem tip. Label the apical meristem, leaf primordium, protoderm, ground meristem, provascular tissue, axillary bud, leaf trace, and leaf gap. Focus on cell density and tissue organization rather than fine detail. Use your microscope observations and background knowledge to answer each of these questions in your lab notebook. Compare the undifferentiated meristematic tissues with the mature tissues they produce. These include the similarities and differences between the epidermis and the protoderm, the ground meristem and the pith and cortex, the provascular tissues in the xylem and phloem, and a leaf primordium and a mature leaf. Focus on cell size, wall thickness, specialization, and function. The apical meristem is the engine of primary growth in stems. Through continuous cell division and differentiation, it produces the dermal, ground, and vascular tissues that make up the mature stem. By examining a stem tip, you observe the earliest stages of tissue organization and how complex plant structures arise from simple, rapidly dividing cells.